Microscope is a tool that we're going to use quite a bit throughout the year. So let's take a look at the types of a couple of scopes. We use a compound light microscope, which means that it's got lenses and a light, and the electron microscope we'll compare it to is something that we don't get to use, unfortunately, because they're very large and they use different technology. So under a light microscope, we can actually see living or dead organisms. It uses light. It will magnify up to 1,000 times, but the ones we're using will only go up to 400 times the magnification. And when you're writing the magnification for the microscope, when you look under it and are drawing a specimen, make sure you always put this X there. That's the unit that you need to have for magnification. You can see quite a bit, but the detail is considerably low compared to our electron microscope, which we'll look at in a minute. And there's a couple of types: the compound microscope, which we're going to use and we'll look at next, and then the dissecting microscope, or what's called the stereoscope. And it also uses light, but the orientation is slightly different. The compound light microscope has a much smaller working space and will show the image upside down and backwards. So there's two different orientations there. And a dissecting microscope will actually keep the orientation the same. The image under light microscopes are 2D, but they are in color. We can use staining techniques. We'll use methylene blue to actually stain a specimen so that we can actually see the organelles a little bit clearer. We can look at cells and bacteria, anything that's going to be less than zero point 0.2 micrometers. Unfortunately, some cons are, there's limited detail and that specimens, do have to be small, and they also have to be very thin so that light can pass through them. And these microscopes are less expensive. The electron microscope, we have to see dead organisms, and it's going to use a beam of electrons instead of light. It's going to magnify 100,000 times, which is incredible. So you can see things really close. So the detail is very high. There's the scanning electron microscope, which is called an SEM, which is used for viewing the detailed surface of cells, and a transmission electron microscope, which is a TEM, used for viewing the detailed interior of the cell. So think of scanning as surface. The S's match up. The image is going to be in 3D, but it's going to be in black and white. There's no techniques that you can use for this, but you can look at organelles inside of a cell, so we can see things like mitochondria. You can see viruses and bacteria. It is expensive, and you can't study living specimens, so those are some downfalls, but it shows great detail and can be used to study specimens that can't be used using a light microscope. So let's take a look at what a compound light microscope looks like. This is similar to the ones that we'll use in the classroom, and we'll label them. So this is going to be the eyepiece, or the ocular, and typically it has a magnification of 10 times. Then we have the body tube. That big knob is going to be the coarse adjustment knob, and the coarse adjustment knob is only going to be used when you're viewing specimens under low power. This is going to be the fine adjustment knob, the smaller knob, and that's going to be used for viewing specimens under high power. Here we have the nose piece, and attached to the nose piece are the objective lenses, and you'll have different magnifications. On each of the lenses, you'll have a marking. It will say 5x or 4x, 10x, and 40x. Now, to find the total magnification of a microscope, you're going to multiply the magnification of the eyepiece times the magnification of the objective lens. So if this lens right now is on 10x, that's 10x, and the eyepiece is 10x. We multiply them together, our total magnification would be 100x. Next, we have the stage clips, which are these little metal things here, which will hold the slide in place. Then the stage, which you put your specimen on. And the diaphragm is actually going to control the amount of light that's coming through. When you move from low power to high power, it actually appears to be darker. So to adjust your focus under high power, you're going to use the fine adjustment knob and the diaphragm. And we have the arm and the base. And you have your light source here as well. Okay, so you'd have a power switch somewhere. So there's a couple things that you need to remember. We always start on the lowest power, which is actually called the scanning objective. And then you're going to use the coarse adjustment knob for the scanning and low power objectives. And then you use the fine adjustment knob and the diaphragm to focus under the high power or the 40 times objective. To find the total magnification, you're going to multiply the eyepiece times the objective lens magnification to get your total magnification. And remember that when you're looking through one of these microscopes, you're actually going to see the image upside down and backwards. And also, everything is opposite. So if a specimen is seen and it's to the right of your field of view, okay, what you're looking at through the microscope, you're actually going to have to move it to the left in order to get it to be centered. And when you move from low power to high power, you always want to make sure that your specimen is in the center of the field of view, otherwise you're not going to be able to see it under high power, because as you increase your magnification, you're actually decreasing your field of view.